Hello and welcome to part 2 of online lecture 6. This is going to be a short part of this lecture, and we're really just going to focus on the discovery of the structure of DNA and the soap opera that surrounded that. So this is going to be a little bit of fun history and also a chance for you to learn that scientists are human beings just like everybody else and do things that are sometimes surprising. So this idea of them being cool, calculated, and objective maybe doesn't always apply, or maybe applies very rarely. As you know, the structure of DNA consists of a double helix that contains two complementary and anti-parallel strands. And a diagram of the structure of DNA is being shown here in this model on the bottom of the page. And you may have seen this picture up on the right-hand side of the page. This is of James Watson, who's in the lower left-hand corner, and Francis Crick, who's in the upper right-hand corner. And these two gentlemen are credited with discovering the structure of DNA and won a Nobel Prize to that effect. And you may even recall that one of their papers was cited in the lecture on DNA replication. And in their structure, they showed that the DNA was actually a simple but elegant structure, that it was composed of 4 kinds of nucleotides. They're joined together by covalent phosphodiester bonds to form 2 long polynucleotide chains that come together in an anti-parallel fashion to form the double helix. So that's the part of the story that everyone knows. This is a somewhat less familiar photograph to people who haven't studied the history of DNA. And this photograph was actually very important in allowing Watson and Crick to propose the correct structure of DNA that was published in the journal Nature. This is a photograph from what's called an X-ray crystallography experiment. And what happens in X-ray crystallography is that a substance is crystallized, and this is used both for proteins and also nucleic acids. And when you form a crystal of something, what happens is that you basically freeze it in a way that the structure is repeated over and over again in a very regular way. And so this is a photograph of a crystal of DNA. And you might look at this and think, I don't really see anything all that interesting here. It just looks kind of like an X in the middle with a couple of black marks on the end of it. Well, you certainly wouldn't be alone in knowing how to interpret this kind of photograph, because it's actually a fairly complicated process to make one. And even when you get a good photograph, it can only be interpreted by a person who has significant training. So I'm going to go through a little bit more of the detail about how this photograph was generated and then tell you why it was so important to Watson and Crick. And the first thing that has to happen is that an adequate crystal has to be obtained. And this is very difficult because the crystal has to be large enough to observe, and also it has to be very uniform. If it doesn't meet both of those specifications, the photograph can be blurry and make it really difficult to actually interpret. So the point of X-ray crystallography, sometimes this is also known as X-ray diffraction, is to determine the arrangement of atoms within a crystalline molecule. And what happens is that once an adequate crystalline specimen is obtained, a beam of X-rays is shined through it. And when the X-ray beam strikes the electron clouds of the atoms in the crystal, the X-rays become scattered. And what's really handy about this is if you actually have a piece of X-ray film in the right location, you can actually collect the pattern of these scattered X-rays. This is precisely what was done to generate photograph 51. And this slide shows the experiment. This is showing a DNA strand stretched across a paper clip and mounted on a piece of cork. And what that really is a DNA crystal that was stretched across there. The X-rays were sent through it, and then you got this X-like pattern. Now, a skilled observer can look at the angles and intensities of the scattering beams and actually create a three-dimensional picture of the electron density of the crystal. And from the electron density of the crystal, then the mean position of the atoms can be determined, and then basically the structure of the molecule can be considered solved. But even with that, one image is not nearly enough to determine the structure of a crystal, so it has to be rotated and different things have to happen. Now, in the case of photograph 51, this photograph was actually taken by a very skilled scientist. Her name was Rosalind Franklin. In May of 1952. She was familiar with working with DNA, and she created this really very clean and the best photograph that anyone had made of the crystal structure of DNA up to that point. Now, Watson and Crick were actually knowledgeable about X-ray crystallography, and so as soon as they saw that X shape on that really nice clear photograph that Rosalind Franklin took, they knew that it was caused by a helical shape of the DNA molecules in the sample. Well, this is a cool story, right? So these guys are working really hard trying to figure out the structure of DNA, and then they get this clue, and they solve the structure, and they go on to win the Nobel Prize. So everybody's happy, right? Well, not so fast. The next slide I'm going to show you has a lot of words on it, and it says free pass at the top. And what this means is I'm about to tell you a little story. I don't expect you to remember all the details of it, but it's very interesting and an important anecdote, I think, about ethical scientific behavior. So let's talk a little bit first about Rosalind Franklin. So she actually died in 1958 when she was 37 years old. She was a Jewish woman who was working in British scientific institutions of the 1950s. You can imagine how difficult working environment were for women just in general back then, and in the sciences even more difficult. And one of the really key pieces of information that allowed Watson and Crick to solve the structure of DNA was that photograph I just showed you. And they saw it because her boss in the lab, his name was Maurice Wilkins, showed that picture to James Watson in 1953 without her knowledge. And Watson's written a book called The Double Helix about this whole process of discovering the structure of DNA. And he said, "The instant I saw the picture, my mouth felt open and my pulse began to race, for he recognized immediately its telltale helical signature." So psychologically, that was the key event that helped him drop everything else and search for the DNA structure. The other piece of information that Watson and Crick used was Franklin's measurements of a DNA unit cell. And so she'd actually measured the size of DNA and was able to include this in a report to the Medical Research Council. And someone shared that with Watson and Crick. And when that was shared with Francis Crick, he realized that the two strands of the DNA helix had to run in opposite directions in order for her measurement to be correct. So the curious thing about this is that without these clues, Watson and Crick would not have been able to put together the DNA structure as fast as they did. Now, they argue that the title of discoverer goes to those who first fit the pieces together. And, and the thing about this is they were criticized for this because they did not include her in the paper that they wrote in Nature. And they were quite relentless. In fact, James Watson was quoted as saying about her, people said, well, you should have given her credit, or Maurice Wilkins credit, for they were nice enough to show you that photograph. And they actually said that nice is what you do when you have nothing else to offer. So what happened afterwards? Well, basically, in 1953, when they published their paper, they did acknowledge being, and this is a quote directly from the paper, "stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M.H.F. Wilkins and Dr. R.E. Franklin and their co-workers at King's College." And they were really being sneaky here, in my opinion, because they really crafted this sentence really carefully so that Rosalind Franklin would not realize what they'd done to her. They took her data, and in fact, she died 5 years later, and she never actually knew that they'd seen photograph 51 and her unit cell measurements. So she didn't know exactly what was being referred to there. Now, her advisor and colleague, Dr. Wilkins, clearly did, because he showed the photograph to James Watson. But this is the part that I think is really galling, and that's that when James Watson and Francis Crick won their Nobel Prize for their discovery, they didn't even mention her in their Nobel speeches. Fortunately, she had passed on at that time. And I say fortunately, so she didn't have to realize just how utterly and completely she had been scooped. By today's standards, she would have absolutely have been a full collaborator and possibly also co-awarded the prize for the discovery of the structure of DNA. At a minimum, she would have been a co-author on their paper that was published in Nature. So I'll pay homage to Rosalind Franklin here by finishing this lecture with a photograph of her. And thank you for your work, Rosie. We appreciate what you've done. And this concludes part 2 of online lecture 6. There's one more lecture to go, part 3.